Okay, so we'll be starting with the week 4 live session. Uh, so as mentioned, so if you have any facing question or anything, you can ask. Otherwise, what we will do is that we will start and you can uh, just a minute. Yeah, sorry. Yeah, yeah. So what we had done was that we had seen. I think you have one all of you have gone through this particular level. So the thing is that we have covered regarding the 2D gel electrophoresis, and now we will be doing for the 2D diet or 2D differential in gel electrophoresis. So now what we will be doing is that first of all, what is the technique? So today is what how we are going to is that to first understand that what was the limitation related to 2D gel electrophoresis, followed by how it has been overcome with 2D dyes or differential in gel electrophoresis, what is the basic principle, what are the dyes that are being used, and finally that how it is helping to have the differential expression analysis, followed by to understand some of the uh, methods for modeling different biological networks and what is systems biology. So we won't be going uh, we'll going to some of those techniques other than in the proteomics techniques other than the mass spectrometry based technique, because from the next week you will have a detail regarding the mass spectrometry based technique. And definitely, as I have mentioned, that once we get time and we are into the mass spectrometry based thing, and with some other hands-on and other things we can have with everyone for uh, further understanding for a particular thing. So yeah, so now we had gone through in detail regarding our 2D daily electrophoresis. So what was the major issues if we talk about? So first thing that was a major issue is that it is the gel to gel variability. But yes, if you have any question, just uh, unmute and interrupt me and let me know. Okay. So now what was the thing is that it's the gel to gel variability, because you have you have to prepare different gels, and the preparation of the gels might take you to have different uh, it would be inconsistent throughout for different samples, and that can lead to variability if you cannot identify the spots well, or if you can't mark it like the landmarks we have discussed that day, that we need to have the landmark spots and identify the actual spots along with that saliency and other things that needs to be changed and accurately done. So definitely what can happen is that you might be facing a certain issue where there might be some variability should be there. Okay. So now the next thing that is the major thing is the limited protein resolution. So 2D gel electrophoresis may struggle to separate the proteins with very similar isoelectric points and molecular weights, leading to overlapping spots that hinder accurate analysis. So this is another thing that can be very much uh, problematic is the resolution, because the that you have seen in the gel, and also I have shown some of those gels with some issues, like if you have much salt or your protein doesn't get solubilized, or there's a certain issues that are being there. And also there you have seen that there are very overlapping spots, right? So these overlapping spots and all would be very difficult if you don't maintain, and that can lead to hinder your accurate analysis. Then is the low reproducibility, that is the gel preparation, sample loading, running conditions. They are all inconsistent outcomes between the experiments, and that will lead you to difficult to compare your results across different runs. Then is the sensitivity. So this is because of the low abundance proteins, and all those cannot be uh, easily find out. So easily it is difficult because of the solubilization and other things that are required. So sometimes what happens is that the detection sensitivity might go low, and it will not help into get the low abundant proteins, right? Now what happens is that labor intensive and time consuming. So the process involves multiple steps: sample preparation, isoelectric focusing. Before that, you need to have your rehydration and all these steps. You can use passive rehydration or active rehydration, and you will be giving all those steps. Sample preparation, then SDS phase, staining, image analysis, all of which are time intensive and require very, very careful optimization. So that's another problem. And then it's that it is difficulty in analyzing hydrophobic proteins. So hydrophobic or membrane proteins often exhibit poor solubility in the buffers used for 2D gel electrophoresis, resulting in underrepresentation or absence in the final gel. Okay, so all these things uh, are there with the 2D gel, though it was one of the very far first attempt to try to find out and try to make an attempt to resolve the proteins with these physical properties that would really lead to have an ideal condition of that to find out and characterize different proteins based on their characteristics like the PI and the molecular weight. So apart from the one dimension, another dimension was used, but still issues like gel to gel variability, limited protein resolution, low reproducibility of variations, sample loading, running conditions, and detection, sensitivity like the low abundant proteins and all, and those who are not solubilized proteins, as well as it is a kind of very labor intensive and time consuming task. And also it was difficulty in analyzing hydrophobic proteins and because of their poor solubility. So all these things are were quite issues. So now what we have come up with another technology, another technique in the gel-based proteomics, if you say for that to say for that matter, is the differential in gel electrophoresis or dye. So basically, what happens is that whenever we want to say that uh, we want to uh, we want to compare, so there is one condition one, and there is condition two, right? So if there is condition one and condition two, so whenever we try to compare, so rather than qualitative analysis, we want to have a quantitative analysis. So what is the meaning of the quantitative analysis? So quantitative analysis is something that we can quantify to understand the differential pattern, right? So this is quite important. If we want to compare condition 1 versus condition 2, we want to have their differential pattern of expression. So these we need to capture and have, so these we need to capture and get it, right? So differential patterns of expression in condition 1 and condition 2. So this is what we need to get it from our particular experiment, whatever we do. So here, this diet is very much suitable. So diet is very much suitable to help achieving in this part to compare the difference in protein expression, compare the protein expression. Now, now we will be looking at how it is done or what are the basic steps and what are the basic principles to which diet happens. Okay. So I'm sure you all have gone through the lecture very carefully. So we are very much will be using psi dyes, of which there are three varieties. If you can see, this is psi 2, psi 3, psi 5. Right. So these are collectively known as your psi dyes. So these are your most commonly used fluorescent dyes in dyes. Why? Because they have this capability or this characteristic property of distinct excitation and emission wavelengths. So these lead us to have simultaneous labeling of different samples for multiplex analysis on the same gel. So the same gel now can be utilized in a multiplexed way. Why multiplexed way? So multiplex means more than one sample. So multiplex means more than one sample can be able to run in a single gel because of these three dyes, psi dyes, that is the psi 2, psi 3, psi 5. Now, any kind of fluorescent labeling, if we want to do in the protein, then we have to also keep in mind that the protein amino acid to that we have to label those fluorescent to the any particular amino acid, right? So, so first of all, these psi dyes are able to attach to amino acids, and second, they have this unique property and capability to distinct excitation and different excitation and emission wavelengths, so that those fluorescent dyes can be utilized to understand that expression pattern. So what are those? So these amine reactive dyes, so these psi dyes are also known as amine reactive dyes. Why? Because these psi dyes are NHS ester derivatives that specifically label your amino acids of lysine residues in proteins. So this psi dyes makes the bond with lysine residues. So this help us to label the protein effectively, because you know that in any protein there will be some lysine residues. So our dye would be able to go and bind to those particular lysine residues in the protein and will give the labeling. And after the labeling is done, since psi 2, psi 3, and psi 5 have different excitation and emission wavelengths, so that would help in enabling you to get different labeling of different samples and will help you into get the multiplex analysis in the gel. Now, among the labeling, so that's what it is written, that ensures consistent and stable labeling while minimizing interference with protein function. Now, protein function process needs to be kept intact, and that should make a stable bond. Those fluorescent dyes should make a stable bond with our particular uh, protein. So here lysine residues, amine reactive dyes, and they have different excitation and emission wavelengths. Now, among the labeling, there are two kinds of terminologies you can see, that is written is minimal and saturation labeling. So this is very, very important topic terminal in the case of dyes, that minimal and saturation leveling, because these are to be used very thoroughly and very rigorously. So how it is done is that this dyes typically uses minimal leveling. So dyes, what is generally used is the minimal leveling is being used to avoid altering protein properties, but saturation leveling can be employed when targeting low abundance proteins, allowing for enhanced detection sensitivity. So minimal labeling is the one which is generally used. Why it is so? So the protein properties does not get changed or altered, because otherwise it will be quite difficult. But then what happens is that there is another saturation level. So if you want to, so one, if you remember, one of the major disadvantage for a particular uh, uh, 2D gel electrophoresis, what we talked about, that it was the major disadvantage was that it was not being uh, it not being sensitive enough to get the particular data. So here what we are saying that it uses if we can use if we to perform saturation labeling, then what will happen is that low abundance proteins will also be getting targeted, and it will enhance our detection sensitivity. So now we will be looking in detail about these two terms, that is minimal and saturation labeling, because as I mentioned that this is quite important for us to understand how it works, why it is important, and how it can affect your experiment. So because in this diet, if you have quite now understood that all the full goal of the experiment is to have labeling it to different dyes and visualizing your particular protein, uh, visualize, get those intensity and try to find out its expression level, and then finally go ahead and try to interpret your result, that what is the case of your particular, what is the case for your particular protein, what is the differential expression pattern. So now what is we are talking about is that minimal and saturation labeling. So what is minimal labeling? So in minimal labeling is, as you go to with the literary meaning, that it is only a small fraction, that is your less than 1% of available lysine residues are labeled with fluorescent dyes. So less than the 1%. So you can understand that why the term minimal came. So it came from its literal meaning that only a small fraction will be of all available lysine residues are your labeled with your particular fluorescent dyes, and it will ensure that the protein's biochemical properties remain largely unchanged. Right now, what happens is that next is the maintaining the protein integrity. So by labeling a minimal number of lysine residues, minimal labeling preserves protein function and mobility. So a minimal number of lysine residues, what happens is that minimal labeling preserves protein function and mobility, reducing artifacts and maintaining separation accuracy. Third is the quantitative comparison. So minimal labeling is ideal for comparative proteomics, allowing multiple samples to be analyzed in the same gel with high reproducibility and minimal dye-induced variability. So any uh, so in any kind of this kind of experiments when we are talking about, so we should be sure that we are not going ahead with any kind of technical variability. So these kind of variability should not be, so technical variability should not be introduced in the experiment. So that's why minimal labeling is sometimes is majority of the times quite effective and quite being used for comparative proteomics. So comparative proteomics, if you understand the term, is to compare. So from the term compare, it is coming, that is for comparison between two conditions, right? So comparison between two conditions. So this minimal labeling is used for your particular uh, comparative proteomics, and that allows you to have multiple samples to analyze in the jail and minimal dye induced variability, and it is only less than 1% of the available lysine residues are labeled. Now saturation labeling, so if everything is fine with the minimal labeling and we should go ahead with only the minimal labeling, then why we are being introduced with the another term called saturation labeling, when it is of when minimal labeling can perform the best with can be very much ideal for this kind of experiments, and can be does not alter protein properties, as well as it makes the reduces and does not induce any kind of technical variability and all those things. And why the term saturation leveling? So how what is the meaning now? So saturation leveling again is the same in the literal meaning, like was for the minimal leveling, which is very minimum. But saturation leveling targets all available lysine residues in proteins, that providing higher labeling efficiency and sensitivity, especially useful for detecting low abundance proteins in complex samples. So what happens is that in the saturation labeling, it targets all available lysine residues in proteins, that provides uh, and it will obviously it will help in providing higher labeling efficiency and sensitivity. And this is very useful tool for detecting low abundance proteins in complex samples. Now to have uh, sensitivity detection, so saturation labeling enhances the fluorescence signal of low abundance proteins, making it possible to visualize proteins that might be undetectable using minimal labeling. So what is the trade-off between saturation labeling and minimal labeling? So which one would you choose for your particular experiment? That will depend upon your research question. So while saturation labeling offers greater sensitivity, it may alter protein migration during electrophoresis due to high labeling density, potentially leading to differences in protein separation compared to minimal separation. So saturation labeling which might alter protein in migration during electrophoresis due to the high labeling density. So it may alter the protein migration during electrophoresis, and this might lead to the differences in protein separation compared to minimally labeled technique. Okay, so this is now we would was about the terminology between minimal labeling and saturation labeling. I think it is quite clear now that minimal labeling is that which are getting attached to only less than 1% of all lysine residues present in the protein sample, in doing in the getting uh, bind to it in a very minimal manner. But when we are talking about our particular uh, thing, that is that is for the sensitivity part, then we are have to go to the saturation leveling, that where we are going to attach as many as lysine residues possible. And what will happen is that it will amplify the signal abundances of low abundance proteins as well. So minimal labeling 2D dyes, saturation labeling 2D dyes. Okay, so what are the differences and in the experimental strategy and how it is done, right? So we have a protein. So as you know that each and every experiment will have to be first optimized, will have to be first understood the protocol, that how it is getting reacted, and then finally we will get the final workflow or major workflow and the experiment. So if you have the protein, you have the lysine residue. So first we will look at the minimal labeling workflow, and then we will look at the saturation labeling workflow. So minimal labeling workflow is that the protein, there is lysine. So you have the 50 microgram protein per 400 picomole dye. So this is the range for the minimal labeling. So that less than 1% residues, lysine residues are. So less than 1% residues are going to be attached with the dye, right? And the dye forms through uh, through the lysine residues, as you know. So this dye, so we are keeping on pH 8.5, and it is 30 minutes on ice, and then quenching of labeling reaction with excess lysine. So you need to quench your excess reaction by giving lysine, additional lysine. That will taken away take away the excess thing. So dye labeling of 1 to 2% of lysine residues. Protein is added with dye, stoichiometry labeled protein, and then we are having the 2D gel separation followed by differential 2D spot pattern, and then the analysis, statistical analysis or proteomic analysis to understand that which particular proteins are having a different expression in a particular individual into different conditions, right? Now, sorry, another labeling, that is the saturation labeling. So you can see that this is used for labeling the cysteine residues generally, not the lysine ones, as it is the case for our minimal labeling. And in the saturation labeling, what it is there is that they don't. So there is no any cutoff or threshold for which we need to make a balance so that we can go for certain excessive. Yes, so we can go for excess dye. So this is so these are excess dye which are being used for getting your protein, right? So if we attach the protein with the cysteine residues, not with the lysine ones, and we are providing excess dye. So please keep in mind for your minimal labeling and saturation labeling. One you are adding with the lysine, another you are targeting cysteine, one you are giving very fixed and limited amount of dye, another where you are giving excess dye, right? So you have pH 8.5, 30-minute quenching of labeling reaction with excess lysine, and you are giving here saturation labeling, and you are not quenching any excess as well, and you are giving excess dye rather. Complete dye labeling of all cysteine residues, dye labeling of 1 to 2% of lysine residues. Now what you are doing is that protein and dye, the substoichiometrically labeled proteins, 2DG separation, differential 2D spot pattern, proteomic analysis. If you say about dye, protein dye for the cysteine residues, so here you can see that there are two proteins, two residues are shown because to imply or signify that it is your saturation labeling. Okay, so that is the major difference of showing this to the significance of it. So saturation level proteins and 2DG separation, differential 2D spot pattern, and then finally proteomic analysis. So one will have your sensitivity. So this will have your sensitivity, low abundant protein, and here is that no alteration of protein characters, and also we are not not adding much technical artifacts. Okay, so this is how this level leveling and saturation leveling works, right? So now, what is the chemistry? So we have understood that we can attach our dye with certain percentage. It is less than 1% for minimal leveling, and whereas for much greater extent for the saturation leveling, or we can add in the lysine residues, or we are writing in the cysteine residues. So basically, how it happens, right? So cyanine dye core structure. So what we have told, the cyanine dyes, one is that it can, if we tend to bind any protein or tend to label our proteins, so that should be binding to some fluorophore, and that fluorophore should have some uh, compound, some bonds that can be able to bind to the proteins quite effectively, right? Now, cyanine dye core structures. So we will be looking now into its structural aspect in a to a greater extent. Okay. So what happens is that cyanine dye core structure is that are based on your cyanine dye backbone consisting of polymethane chains containing two nitrogen heterocycles. So this provides you the tunable fluorescence properties. So Psi3 for green, Psi5 for red. So this is that two nitrogen containing heterocycles that provides you the tunable fluorescence properties. So tunable means that some kind of change thing that is right. So this is what is being required to great extent to have the cyanide dyes that will help to get like the polymethane that have the heterocycle nitrogen containing heterocycles that provides us the tunable fluorescence properties across our different wavelengths we will be getting. So that's why the Psi3 can go for green, Psi5 for red, Psi2 for yellow. So all these backbones and these heterocycles, tunable heterocycles are helping us in getting different fluorescent properties. Now the second thing is that what it is asked for is that it is reactive NHS ester group. So the Psi dye molecules are functionalized with an N hydrosuccinimide NHS ester group. So that is highly reactive towards primary amine, means facilitating efficient coupling to proteins via lysine residue, right? So this how it is done. So these are functionalized with NHS ester group. So this is how, this is one very important thing to understand, that this side molecules, what it does is that it has hydrogen in hydroxysuccinimide ester group that is highly reactive towards primary amines, facilitating efficient compared to proteins via lysine residue. So this is what their major thing, that it is being functionalized with NHS ester group. Third is the amide bond formation. This NHS ester reacts specifically with your amino group of lysine residues in proteins under mildly basic conditions, right? So that's why we have said that it is on pH 8.5. So if you see, it is pH on pH 8.5. That is mildly basic condition, not fully or not very roughly. It's just a mildly basic condition. So in mildly basic condition, what it is happening is that we are keeping the reaction on, and we are in hydroxyamide ester group, which is quite relevant and highly reactive towards primary amines. So it is facilitating efficient coupling of proteins via lysine residues. Then this NHS group ester, amide formation, then minimal structural interference, a chemical device over design of side eyes ensures minimal addition so to the protein's overall mass and charge. So it gives a really minimal addition of the protein's overall mass and charge, and it preserves its native electrophoretic properties and ensuring accurate separation during 2D gel electrophoresis. And optimized spectral parameters are there. The polymerase gel, the polymethylene chain length and substituents are determine their excitation and emission, allowing for multiplexing with minimal spectral overlap, critical for accurate dye analysis. Right. So, what are those techniques? So, one second. Yeah. So, where we were? Yeah. Yeah. So, where we was that we have this sand and dye port structure first of all. So, which are enabling us to have this side eyes where we can have a cyanide dye backbone and consisting of this polymethane chains, which contains two nitrogen containing heterocycles and provides tunable fluorescence properties across your different wavelengths. So that is the psi 3 for green and psi 5 for red. Then it's the reactive NHS ester group. So the what happens is that this particular side dyes are being functionalized with an N-hydroxysuccinimide NHS ester group that is highly reactive towards primary amines and facilitates the efficient coupling to proteins via lysine residue. Now, if we see for the amide bond formation, so this basically reacts with the amino group and forms the amide bonds under mildly basic conditions. So that's why I was showing you that it is for the 50 microgram protein and those dye, which is for 58.5, that is your mildly basic condition. And what you are doing is that you are getting it in the with the excess lysine. Now, what happens is that it specifically with the amino groups after it is done, so we have the minimal structure interference. So this ensures there are minimal addition to the protein's overall mass and charge, preserving its native electrophoretic properties and ensuring accurate separation. So there are reduced technical artifacts. And then we have the optimized spectral properties. So these guys, their excitation and emission wavelengths are different and it is qualified critical. So now we are talking about the labeling chemistry. So we will just click on it. Yeah, so sorry, just continue. Yeah, so here what you can see that this is the proteome A, proteome B. So if you, so we have said that in a single gel, so to reduce the gel to gel variability, so that was one of our concern when we talked about 2D gels limitations, 2D two-dimensional gel electrophoresis dimensions. We talked about that case. We are not very much familiar, not very much ideal condition that the gel, if we are running each sample in different gel, then there might be highly technical variations, and the variations can be, we cannot able to understand whether it is for the biological variations or the technical variations because each sample is running in different gels. So what we have said is that we have the Psi3 and we have the Psi5. So this, as we have mentioned, that these have some heterocyclic nitrogen groups that gives us the difference in particular excitation wavelength, minimal spectral overlap. So what we are saying, this proteome A, we are matching from one condition, that is coming from one condition, that can be from one patient, that can be one disease, control, treated, anything, what it can be anything. So this proteome A, we are adding with Psi3, whereas proteome B, we are adding with Psi5. And then what we are doing is that we are pulling those two proteins. So this is the NHS reactive ester group. So this is what we were talking about. So this is the dye, this is the protein, and this is the NHS ester reactive group with pH is 8.5, 0 degrees C, 30 minutes, and protein with the dye. So this is quite important process and procedure. So now how this dye works, right? So this is what is our experimental strategy that we generally take. So what is our general experimental strategy that is being taken? So one, so for example, this is with the plasma sample. So we can say that this is with the plasma samples, right? So this is with the plasma sample. So you can try it with any other specimen, any other sample that you wish to. So what they are saying is that depleted plasma 1, depleted plasma 2, pooled internal standard. So first of all, can anyone of you tell why it is depleted plasma? So because what happens in the plasma, in case of plasma proteomics, so what happens in case of plasma proteomics is that what we are getting. So what we identify is that there are abundant proteins. So what is the abundant protein? We are, so you know that blood, plasma or serum are highly enriched with some proteins like albumin, you have transferrin, and all those proteins, to 12 to 14 proteins are only very, very, very abundant that mask each and every other protein. Okay. So these depleted plasma is that they will remove these proteins who have a significance and have doing of lesser abundant proteins. So what we want to do is that we want to have a lesser abundant proteins that can be the actual markers, whereas that will get masked because of this kind of abundant proteins. This is the abundant proteins like albumin and others. We will not get the exact signal, right? So we want to be getting our exact signal out of it. So for that reason, what we are doing is that we are depleting the samples. So depleting here means just to get rid of these highly abundant proteins so that the lesser abundant proteins also get a chance to come and to we can measure their intensities. So now the depleted plasma 1 is labeled with Psi3, 2 that is the conditions. One condition is with Psi3, one other condition with Psi5, and the pooled internal standard with the Psi2. And then what we are doing is that we are taking the picture and finally we are overlaying for each spot. So what it is there? So what we are talking about is the mixed labeled sample 2D separation, then followed by image each gel at three different wavelengths, Psi2, Psi3, Psi5, sample 1, sample 2, and pooled and image analysis. Okay, so hopefully it is clear for everyone. So now what we are talking about, what we are talking about now is that so now we have talked about the experimental strategy. So we know that the dyes are labeled to these three different. So these three different groups. One will be going with the Psi5, one with the Psi3, one with the Psi2. So now one is the one which has, for instance, we have our samples for condition 1, sample for condition 2. We are adding it, mixing it. We are labeling them with Psi5, Psi3, and Psi2. And then what we are doing is that we are analyzing it in different wavelengths because of their fine tunability and have to have in different wavelengths. So we can easily go ahead and uh, get the uh, scanning done. So the conditions are Psi 3, Psi 5, Psi 2, and then we are going. Now what we will do is that I think so all of you have noticed that one sample condition we are measuring with Psi 5, another with Psi 3. But what is this third thing that we are getting with Psi 2? So that is our major question or major understanding if we are trying to understand the dice experiment strategy. One, we have understood the labeling, the labeling efficiency, and we have the minimal labeling and the saturated labeling. One, minimal labeling because it helps us in not altering the protein structure, whereas saturation labeling is one which alters the protein structure, but it is highly efficient and sensitive for lower abundance. So now, what is now if we try to understand in a deeper manner, so why there are three dyes we are using when the conditions are two, that is only one, and we are trying to match with one condition with another condition. But what we are saying is that we are having three different dyes and we are adding three different conditions. So now what is the case? So case is that label condition A with psi 3, label condition B with psi 5, so mix condition A and B that will make the pool of internal standards, right? So how this is done and why pools are required? So what we told that label pools. So for instance, what it is the meaning of this? So for example, condition 1, condition 2. So these are 5, tools. One, two, three, four, five. Right. So now what we will do is that we will take wall amounts. So these wall amounts from each of those tubes is collected in one tube. So this is labeled with Psi2. So why this? This is the pool sample. So why this pool sample? What is will be this work? So we are saying that these pool samples will be used to normalize and understand the gel to gel variation. Why? Because this is the same sample. Just a minute. Yeah, just a minute. Sorry for this technical issue. So what? So what we were saying that this is only one sample, though it is made from other samples, but in this pool now it is only one uniform sample. So if you mix it and then you do vortex and homogenize it, so this is one solution. Now this is run along with this site 3 and site 5. This site 2 will consist of the protein spots which is present across uh, irrespective of the spectral level because it has got now mixed. So now when we are running the gel, so this Site2 would be helping to normalize it because that's the uh, same sample. So now you will have, for example, you will be there are 10 samples, so there will be 10 gels. So why 10 gels? Because one gel will contain condition 1 and condition 2. So like this, not 10 gels, so five gels is what required. And in all these 5 gels, this from this pool, it will be getting added. So there were 5 gels that we are running for analyzing 5 samples of 2 conditions each. So that is 5 from condition 1 and 5 from condition 2. So now how we are going to analyze is that we are going to analyze through a particular Site2 dye, that is the pool sample that we have made. So that sample will go on in each gel with the Site2. So now when we will be aligning it, so if there are any gel to gel variation, since Site2 is the one Site2 dye labeled pool sample is the one that shows that it is the same technical replicate, same technical sample. So all the proteins in that particular spot would be should be present in particular spot only. But what is happening? So what is happening is that what is happening in this particular spot is that whenever we are getting in this particular spot, then the thing is that so now if there is any gel to gel variation, since that is coming from the same particular sample, then what will happen is that we would be able to try to align those spots and will be normalizing it. So this is how it is done. So sample 1 psi 3, sample 2 psi 5, standard psi 2, and the gel. So since it is the full samples, as I just now mentioned to you all, that what we are doing is that we are getting the exact same sample. So this is from the same tube. This is the full tube, and this is also coming from the same full tube. So this should come, each spot should come at the same place. If it is not there, then there are gel to gel variation. So that needs to be overcome by aligning those spots, right? So by aligning those spots, so those spots needs to be aligned so that what happens is that we get an exact uh, so we get an exact replicate, exact understanding, and we can normalize the other spots as well and reduce the technical variation. So this is how things are working, how things happen in our diet multiplex analysis. So this gives you an broad overview of how things happen. So now let us go through individually through each particular step. Okay, so that we should do is that we should go ahead and have those particular steps one by one, right? So that we will have to go each steps individually. So here they have given very in a right manner how it is done. So each sample labeled with a different side I, side 2, side 3, side 5, combine into 1, 2, combine subject to 2 detailed electrophoresis. So side 3, side 5, and side 2, that is the side 2 is the full, side 5 and another condition, side 3 is another condition. So all these things are led to what? So that it is pulled into it one tube, then subject to 2 detailed electrophoresis. So one is that it will be going through your particular isoelectric focusing. Second is that what we are doing is that separating through molecular weight. So this helps us to get the image at lambda specific for each side eye, and then it will be overlaying the images. So this image analysis helps to identify the differentially expressed protein spots, and then finally you can identify those proteins through different multi-top or any other image-based techniques, where you will be getting that particular thing that is that you are getting a particular uh, the proteins that you are getting that are differentially expressed, and if you want to identify and characterize those particular proteins, basically what you can do is that you can go ahead and do any kind of image based technique. So multi-top image is there for some basic things. So now this kind of images after overlaying and after normalizing you will be getting. So now you need to mark your borders, and you need to, if you remember from our previous 2D knowledge, that we need to mention like those which are present, right? Like our saliency, that which one is particular noise, which one is particular thing. So all those things, and then finally we need to get the intensity out of it, the expression level out of it, and finally correlating with it. The intensity is correlated with the expression level, and once we are sure of the expression level, we will be knowing that which particular proteins are differentially regulated, that we are going to take it forward for our further analysis and characterization as mentioned. So how this data analysis is done, if we talk about a little bit. So image acquisition and alignment. So after 2D gel electrophoresis, that is after we are getting separating it in one dimension, and then after addition of dyes like the Psi3, Psi5, and the Psi2, and then what we are doing is that we are after uh, aligning that after 2D gel electrophoresis is done, that is for particular first it will be separated through the basis of a PI, and then we with the molecular weight. So it will happen is that, so these fluorescent images of the gel will be captured for each dye, that is Psi2, Psi3, Psi5, using a multi-channel scanner. So this kind of scanner will be helping you to capture these three different dyes, different because of their characteristics of different excitation and emission wavelength. So this will be utilized, and then what will happen is that then images are then aligned to correct for angel distortions and ensure that will ensure accurate spot matching across your samples. So it will be ensuring the spot matching across the samples. Spot detection and quantification. So software automatically will detect protein spots on the gel and quantifies their fluorescence intensities, and the intensity of each those spot will reflect your relative abundance of the protein in each labeled sample. Then you have the normalization using internal standards. So internal standards which are labeled with your Psi2 is used to normalize. Again, the concept remains the same. That since this is a technical sample, same technical sample with a homogeneous protein mixture, so therefore its samples, protein spots, and the expressions should remain equal throughout. If it is not, then there is something problem to it. Then what we are doing is that we will be normalizing those for the spot intensities and our different gels, and then we will be doing the comparison of protein expression levels between the samples. Next is the statistical analysis for differential expression. So after that, some basic statistics will help us understand that which particular proteins are significantly altered, are significantly we are getting, and which ones are no. So this is help us to get for the statistical analysis, right? And then what we can do is that if you want, we can go to have an identification by a mass spectrometry based method that actual which particular protein is that, or particular spot is the one which is showing so much differential regulation. So these are the basic 4 to 5 steps that are uh, being employed whenever we want to perform the analysis of the diet or the differential electrophoresis. First one is that we have to go ahead and have our particular thing, that is particular image acquisition and alignment that we need to perform. Second is that we have to capture each image of this dye. So Psi 3, Psi 2, Psi 5 using multi-channel scanner. Then we have to correct it, align it, Then we have to spot detect, we have to normalize using Site2 dye because that's why the internal pool sample and the Site2 dye is provided. We have to go for a protein identification via uh, mass spectrometry if you want to do it. And obviously, the statistics is quite important to understand that the whatever expression difference we are observing, that is the difference in the intensity, is either randomly occurring by chance or is actually the significantly differential. So that we need to understand through dye analysis. So what are the advantages? So among the advantages we can talk about is that accurate quantification with internal standards. So dye uses an internal standard on the same gel, minimizing variability and enhancing precision in quantitative comparisons. So in second is the simultaneous multiplexing. So multiple samples up to three are labeled with different dyes and analyzed together on one gel that enables direct and efficient comparisons. Then third is the high reproducibility. So that will running all samples and identical conditions on the same gel. So there is the major thing, right? So what we are focusing to or what we are aiming to say is that just wait a minute. So there's some technical issue. I'll just share it once again. Please bear with me till that time. So sorry. Yeah. So what were the limitations we had talked about? Yeah, so whatever limitations we had talked about in our particular 2D gel, so all those things are somewhat could be rectified, could be made better for the specifically for the quantitative purpose or quantitative specimen. So all those are quite relevant, keeping in mind of the studies. So first of all, the internal standards and its role is going to play a major role, right? So this is what is very important, that it helps us to get accurate quantification with our internal standards, and this uses on the same scale, minimizing the variability and enhancing the precision in quantitative comparison. Next, what it is required is that the simultaneous comparisons, right? So simultaneous multiplexing, so multiple samples up to three are labeled with different dyes and analyzed together on one gel that enables for direct and efficient comparison. Third is that high reproducibility. So running all the samples under identical conditions on the same gel will reduce the experimental. So this is where I was trying to highlight that for 2D, each and every condition needs to be done separately. That becomes very difficult to uh, claim for anything, keeping in mind that the gel to gel variations and those artifacts will be introduced to a greater extent, and how much it is similar and how much it will be effective in comparing becomes very much challenging. But in this scenario, what happens in the dyes is that when since two conditions are at least run in the single gel and there are such five gels, so then it becomes easier for us to compare because we have uh, there is no run to run variation between in each gel between at least two conditions, and they are under subjected under same conditions, experimental conditions, followed by we have our Site2 that that internal standard pool standard for our reference, that what was the gel variation between the five. So intra variation, so there's no inter variation, and so there's no intravariation. Intervariation is also reduced by is also being captured by running an internal standard. So that's why this dice is quite reproducible in the manner, quite reliable in a manner rather than 2D simple 2D gel electrophoresis. Then we have enhanced sensitivity for low abundance proteins by saturation labeling. So we have again talked about in great detail about the two types of labeling techniques that are very much widely used in this kind of experiments for dyes. Is one is our minimal labeling technique and another one is our saturation labeling. So minimal labeling is the one which minimally labels but also keeps the protein function intact. But saturation labeling will not keep the protein function intact, but it will help us in increasing our sensitivity for getting the detection of low abundance proteins, and it will offer greater insight into the complex samples. Then again, that point which we are referring to from the beginning is that reducing or reduced gel to gel variability that will analyzing multiple samples in one gel and eliminates the inconsistencies in comparisons across the different gels. Then we have the robust quantitative analysis that will provide a wide dynamic range and stable signals, ensuring accurate signal quantification. So these are some of the advantages that are there for dyes. Now, if we talk about again, sorry, just uh, bear with me. Uh, Sorry, so there was an issue. We run into some issue. Hmm. Yeah, so we'll be sharing the screen in just a few minutes. Okay, so we are going to with the yeah. So again, now it's there. Okay. So yeah. So now after getting the advantages of dyes, but still we have certain limitations, right? So what limitations does dyes? Yeah. So dyes suffers from these limitations. First of all, it is a complex setup. So it requires meticulous sample preparation and handling, and which can be technically challenging. Sample limitations typically allows analysis of only up to 3 samples, plus an internal standard per gel. High cost, fluorescence dyes and reagents are expensive. Then we have the spectral overlap issue. That issues with dye overlap can affect our quantification accuracy and requires high resolution imaging systems which will not be available in all labs. So these are some of the limitations that are there specifically for dyes, like the complex setup, samples preparation limitations, high cost, spectral overlap, and the specialized equipment that might not be very feasible for all sorts of experiments. Okay, so this is one thing that is quite relevant. Okay, so now before going into this topic of some systems biology, and as I have already mentioned at the start of today's lecture, that we will be also looking at some other techniques, proteomics technologies or techniques apart from mass spec and the gel based, what are used for different applications. So before that, we will look into some of the things that are also quite relevant in that what is the role, practical application, and particularly this kind of dyes and 2D dyes where are used. So there is something called HPPs, host cell protein. So what are those? So first of all, we will let look into uh, this thing first, and then we will try to understand how 2D dyes and what are the different other applications and other advantages that are being uh, that have been done, innovations have been done in these technologies and are being used in the industry. So if it is sound is thing, so 1 second I will. Uh, I think now it will work.
Two different species in a single cell. For example, the gene for human insulin can be inserted in different species.
Okay, so but I think the video is also self-explanatory. If you
In a
don't
single
understand,
cell.
then you can
For example,
get
the
it.
gene for human insulin can be inserted into bacterial cells to make the bacteria produce insulin.
So what you can do is that I can make it a
Today,
little bit
many
slow for
medicines are
you.
created using recombinant genetic technology. These high-tech methods
Right? Yeah. So that
combine
would help you to
DNA
grasp it.
from two different species in a single cell. For example, the gene for human insulin can be inserted into bacterial cells to make
It's
the
showing about the insulin, how
bacteria
protein can be done through cloning.
produce insulin. When medicines are made this way, they're called biological drugs. Over 70% of biological drugs a made in Chinese hamster ovary or CHO
They say most biological drugs are
cells.
made in mammalian cells.
Others are made in the bacteria E. coli. When biological drugs are produced using recombinant technology, the result is a mixture of the desired drug and unwanted impurities. Some of these impurities are host cell
So there's after you plasmid and if
proteins
you do the recombination, so there is the
or
drug substance as well as the host cell proteins.
HCPs. HCPs are a result of cell stress during the drug manufacturing process. Recombinant cells can function in unpredictable ways, or even undergo lysis or splitting. Some impurities are easily removed.
So this is the drug substance.
However, HCPs can be very similar in biophysical
And C, the
properties
drug substance is a
or
contaminated cell of host cell protein.
non-specifically bind to the intended drug substance. This makes them difficult
Just a
to
minute. Yeah. So I think yeah.
eliminate.
So these HCP are the contaminants present along with your drug substance
HCPs
that was
can
intended for you to
often
make.
evade the
So you can see that
normal
latest of
drug
inoculum,
purification
bioreactor, centrifugation,
processes
filtration,
used
then liquid
by
extraction, formulation, lyophilization, quality
manufacturers.
control, and packaging. So this is the
They're
complete pipeline
sometimes
for your
called
biopharmaceutical if you want to do it in a
hitchhiker
production.
proteins because they can hide among the desired substance. Unfortunately, HCPs that make it into medicine can be a problem. Even small
Okay, but
quantities
see, there's a
could
problem. This red thing is
trigger
the
a
contaminant from the host cell protein that is
serious
being there.
immune reaction in the patient or interfere with the way the drug works.
Okay,
Many
so that is the sign of danger.
HCPs are enzymes. These can be a problem especially when it comes to drug stability. As much as possible, HCPs
You see, these all
must
contain the
be
contaminants of the host cell protein.
removed from the drug. The finished substance should be as pure as possible.
An
Cygnus
HCP-free substance is the goal.
Technologies is investigating new ways to identify HCPs in biological drugs. Pharmaceutical companies invest huge amounts of time, effort, and money into developing new drugs. Part of this is making sure that HCP levels are as low as possible. Companies that make biological
But when
drugs
you are going to,
must
so after
report the
all those
levels
kind of
of
heavy investments, when you are
HCPs in
finally
their
trying to see
substances.
your packaged biopharmaceutical that
Regulators
you are thinking is it
require that
reaching the market,
HCP
but what is
level
the
in
issue?
biological drugs is below 100 parts per million. However, companies decide themselves on an acceptable below 100 parts per million level and extent of characterization of HCPs present in their drug. Ideally, companies should continuously monitor levels of HCPs in their drugs. This helps to show that their purification methods are
Right? This is
consistent.
how we are doing.
Companies should not treat HCP reporting as simply a
So now I think
box-checking
all of you have got a
exercise.
very good glimpse of what is
Cygnus
host cell protein.
Technologies
Now what we are
encourages companies to aim to ideally as past the
how we are going to do. So
purification
that is what
process will
simply is
help
fascinating us more with
researchers to
the technologies that
improve
we have
their
understood, like the 2D
methods.
gel and 2D dyes. How
Ultimately,
this particular technology is
reducing
going to
HCPs
be
leads to better, purer
utilized,
medicines
right?
and a
For
lower risk for patients.
so this is
So what is
what
ClickUp? ClickUp is an
required
all-in-one
to manage
productivity
basically
software where you can manage
and
everything and
enhance your
anything
route
related
to
to your
market.
work. Your
So
documents, your
making
chats,
biologics,
your
I think
whiteboards,
all of
your
you
projects. The
have
value of
understood
this,
it
you don't have to buy
carefully.
5 softwares to do this. You don't have to be hopping back and forth between multiple tabs trying to find that email from a year ago. Instead, you can find
I think
everything
all
in a
of you
matter
have
of seconds,
really
keeping your work and
understood
everything related to productivity
it in
organized
a
in a single place.
that what is the challenge of
Do you
having
know what will
HCPs,
happen
because
if you
that will
read
contaminate your
one book every
biopharmaceuticals, and
week?
after a lot of
Let me share
investment and
some three
and
powerful
after a lot of
benefits.
hard work, when you are going
First
to
one,
launch your
you will
product
improve
after
your
pruning,
knowledge and
getting
ability to
it
solve any of your
synthesized and
problem.
everything,
Point number
then you
two,
will be
you will reach top
getting the
1%
contaminants. So that
doers.
is not
All
very
successful
much
people across the globe, they have
detrimental.
one thing in common.
So if
They read
my
books, they
HCP
read
assay
books,
good,
they read books,
ELISAs are the
especially
most common method.
non-fiction
So
books.
ELISA,
Point number three,
are the
You will
most common
unlock
methods, but
magical
they
power
rely
of
on
your
affinity and
mind and
selectivity of
attract
antibodies, and those properties
ideas
can
that
vary.
will help you grow
So you are unlikely
exponentially.
be to able
But
to
how to
detect
read
every
one book
HCP in
every
your
week?
sample.
Average
Okay,
reader reads at
so
150 to
HCP ELISA
250 words
coverage
per minute.
should
However,
be used.
speed reader
So US
reads at the
Pharmacopoeia has
speed
compared the
up
means
to
of
800
2D
to
to
1000
their
words per minute.
terms,
So far,
that
I have
you are
helped
understanding
thousands
that
of people
whether
to
your
achieve
2D
this goal.
SPS
If you
page
are a
and
working
those things
professional, are
are
a
not
business
being very
owner,
much
trainer,
relevant
or coaches, or even
to this stage,
homemaker,
but it is
if you
very
wish
much
to
relevant because
achieve new
US
heights
Pharmacopoeia
by increasing your
has
reading speed and
compared the
retention
merits
ability,
of 2D
I'm
SPS page
inviting
and
you to
Western
join
blot with
my 6
immunoaffinity
days
binding
challenge,
or
reading
2D
challenge.
SPS page,
And
but
you will
more
be
complex
learning
approach.
how to
So
double your
these are
reading
the two
speed
methods,
within 6
2D
days,
SPS page
how
and
to
Western blot,
retain
which
any
provides
information
you good
quickly and
separation
easily.
of HCPs.
And the third one,
That
how to
requires
read
two tips,
one
making
book
spot
every
matching
week.
challenging,
Just imagine
calculated
if you
for.
read
So
one
this
book
is
every week,
other forms,
52 weeks,
that variable
52 books.
transfer
And
and
even if
those
you
things, and
pick
then
up
the
just two
immunoaffinity
key
binding
takeaways from every
or
book, you can
2D
transform your life. Additionally,
SPS
you
page,
will develop
which
powerful
finds
morning
HCPs
habit as
under
a
native
bonus.
conditions
So
and
please
in
click the
cell
link below
imaging
and
avoid
register
challenges of
my 6 days challenge
membrane
to
transfer. So
see
this tight
amazing
HCP
results.
antibody binding
So
is
please
through
click the link below
underestimated
and
coverage,
join. This is Dr. Manjunath, I'm a
inconsistent,
speed reading
and
and mind
not as
performance
sensitive.
coach. I'll
So
see
this
you
is
inside
affinity
my
and diet
masterclass.
versus
Thank you so
2D
much.
SPS page with plot for HCP coverage. So I can share this thing with the link for you all to go ahead and read it.
Mix and go
Okay.
ELISA kits are designed to
So,
perform
so
with
then here is the
most
dice
natural and
in blot electrophoresis.
recombinant
So
protein
both
A
affinity dice and 2D
constructs
SPS page,
and
and this moving and
incorporate
layer are
the
related to how well are
revolutionary
your
single
enhanced for
step
cell. So you
sample
can go ahead and
treatment procedure.
read it according as
These
I have given you the
Cygnus
link. So you can
Technologies
go
kits
there and
also
go and explore it
reduce the
and have
complexity of the
all those things,
assay,
whatever
as well
interest
as
you can
time to
go
result,
ahead and read about it.
all while
Specifically, these are
increasing
all
the
new techniques and
robustness
advancements that
of
is
the
being done in this
method
field with
over
the particular
traditional
uh,
protein A
principles.
ELISAs.
So here you can see this is the
For
2T
first
differential in
time
blot electrophoresis,
users,
2T diet
we
uh,
recommend
from the
trying our
binding and
mix and go
variant or improved
kits
version of
as
2T
they
diet.
are the most
You have labeling with
robust
HCP with
and
type 3
easy to
isolysis superfin,
use
initial transfer
protein
to membrane
A kits
immunoprecipitation and
on the
wall-to-cell methods, image
market.
analysis workflow. So you have the quality
The
control, alignment, spot
F610
detection,
uses
detection, edit, and
standards
presence filter coverage,
calibrated to
coverage
unnatural
results. Then you
constructs
have the
such as
reproducibility
map
and all those things.
select sure.
So this you can go and
While the
understand in great detail. So this is
F600
a poster
uses
mentioning
standards
about the 2D type.
calibrated to natural
Now we'll
and
see another
conserved protein A.
uh,
Host cell protein ELISA and
basic thing,
HCP
that is
antibody
how
coverage analysis.
Cygnus
An
Technologies
ELISA, or
also
enzyme-linked
offers
immunosorbent
when
mix
assay
and
the
test
go
kit,
cells
uses
kits
get
for
hot.
specifically designed and
protein
reactive
A
antibodies to
I
detect
will
and
resins
just say this
from
thing so
quantify
that you can
host
JSR,
cell
understand
protein
it in much
or
TOSO,
detail.
and
HCP
Kaneka.
So
impurities
ELISA,
found in
how it is used. So this will give you an
biopharmaceutical
This
understanding
drug
that what
video will
particular
substances.
demonstrate
techniques are
proper
used
The
for
techniques
identifying
identification
for
and
HCPs.
monitoring
performing
of
So
a
identity
mix
critical
HCPs
and
and
is
go
functional with
critical
assay.
ELISA
to
or
ensure
enzyme
Let's
experimentation, one
purification
begin
of
by
the
process
reviewing the
quite
sample
how, as I
consistency
have shown you,
treatment
and final
and you can go
procedure.
drug
through as well
substance
on your own in that
purity.
particular article that
First,
how
The capture and
can
prepare
change
the
and
detection
initial
another technique,
antibodies
sample
another
found in
uh,
most
dilutions
evolutionary
ELISA
as
kits
required
states
usually only
prior
test
to
for
couple
sample
one or a
treatment.
few proteins at a time.
So this is
But
for our
the
food protein
antibodies found
analysis.
in
Then
the Cygnus
transfer them
HCP ELISA
to the
kit are different.
appropriate wells
They're
of the
broadly
sample
reactive
treatment
and can
plate.
measure hundreds of different HCP impurities in a single assay.
All
But before we
subsequent
get too far
dilutions to
ahead,
be
let's walk
assayed
through
can
how
be
this
made
process
in the
actually
sample
works.
treatment plate
The
using mix
assay
and go
itself only
sample
consists
diluent.
of two main
You will
steps.
need to
We can see
ensure
now
First,
that
I
the
am showing you
final
add
to have a clearer
volume
idea
in
regarding the
anti-HCP
every
proteomics.
well
antibodies
is
conjugated with
100
HRP
Happening
microliters.
that
enzyme and a
we will
test
understand that how it
sample
works and how to be died
containing
or to be died
Now,
HCPs
with
or
add
HCP
100
accurately and how
standards to
microliters
the
to
of
work
the
with them
well
kit
will
coated with
standards
work according to
and
that idea
anti-HCP
or
controls
understanding.
antibodies.
to the wells.
Cover and place the plate on an orbital shaker for 2
Then,
hours at
add
room
50
temperature.
microliters of
Then
mix and
wash
go
the plate and do
denaturing
a
buffer to
colorimetric
each
detection
well
with a
and
TMB
immediately
substrate for
mix.
30 minutes.
For
Afterwards,
example,
add a
if the
solution to
replicates
stop the
are in
reaction
A1
between
and
HRP and
A2,
TMB.
position the
That's it.
multi-channel pipette
As you can see,
horizontally
the test
when
does not
adding the
require
mix and
special
go
expertise to
denaturing
run.
buffer.
It's also easily
Mix by
transferable,
pipetting up
reliable,
and down
robust, and
approximately
effective in high
15
levels of
times.
protein.
For best
However,
results,
HCP
mix
ELISA
quickly to
assays do
avoid
have some
creating
limitations.
bubbles in the
You'll need
sample
additional
treatment wells.
orthogonal methods to get a complete
It's
story.
very
And the tests
important
are
to
only
mix
as good as the antibody
immediately after
used.
each
Which
pipetting
brings us
action.
to the next part of the
For
story,
example,
coverage analysis
add
of
the denaturing
anti-HCP
buffer to row
antibodies.
A
The
and
wider the coverage,
immediately mix.
the more reactive the
Then
antibodies
move to
will be
row
to
B
HCPs
and
in your cell
repeat.
culture process.
Use
The
fresh
traditional
tips
coverage
for
method
each
is a
addition.
2D Western blot
Blot
compared to a
and
silver
silver staining.
stain.
And
This
then
process
incubate
shows
on the
coverage
bench
through
for
counting the
5 to
unique
10
spots that
minutes.
appear in both the 2D Western blot and the silver stain.
Next,
Unfortunately, this
pipette
method lacks
100
sensitivity and
microliters of the
specificity.
anti-protein
Cygnus
AHRP
developed a
detection
superior
antibody
antibody
into
coverage
each
method
well of
called
the
antibody
antibody-coated
affinity
microtiter
extraction,
plate.
or AAE. In this procedure, ELISA antibodies are covalently
Then,
immobilized on a
pipette
chromatography
25
resin.
microliters of the
Samples are
denatured
then passed
standards,
over the column packed
controls,
with that
and
resin to
samples into
extract
wells
immunoreactive
indicated on your work
HCPs.
list.
The HCPs are eluted from the column, buffer exchanged, and
When
concentrated
finished,
to the original
cover
sample
the
volume.
plate.
The sample is then compared to HCP in the initial sample with a
And
large
incubate
format
on an
2D
orbital
gel
shaker
detected
at
by silver
400 to
stain or
600
2D dyes,
RPM for
and an
1
image
hour at a
analysis is
room
performed
temperature
to
of
match
24
spots.
degrees Celsius
Comparing
plus
these
or
results
minus
to the
4
results
degrees.
found with a 2D Western blot coverage
After
assessment
1
shows
hour,
both an
dump the
increased
assay
sensitivity
plate
and
into a large
specificity
sink
when
or
using
appropriate
AAE.
container.
The column is
And
also
then
run under
blot
more
the
natural
assay plate
conditions.
over
So
absorbent
AAE is
paper.
better predictive of how
Be
the
sure to
antibodies will
gently
perform in
but
the ELISA.
firmly tap the assay plate
The
to
antibodies
remove
found
most
in
of
the
the
Cygnus
residual liquid.
HCP ELISA kits have had
Aggressively
their coverage
banging the
assessment
plate
tested
or
with
the
both of
use
these
of
methods
vacuum
to ensure our
aspiration
kits
devices
detect
to
protein
remove all
impurities with a
residual liquid is
reproducibility that
not
supports
necessary
regulatory
and may
compliance.
cause variable
Have
dissociation of
questions or want to learn
antibody-bound
more?
material.
Visit our
This
services
can
page and
result
speak
in
to an
lower
expert.
ODs and
for
less
you to
precision.
learn and get benefited. So thanks to
Next,
them.
fill
So
the
now what we would
wells
do is that we will just
generously,
quickly move into the next part and
overflowing
try to
them with the diluted wash solution. You
complete
can
the
do
today's
this
session.
by
And obviously, if you have
using
any
a
doubt, you can
squirt
ask, and then we
bottle
can
or by
go to
pipetting
our
in approximately 350
quick
liters.
session.
Then
So
dump the
problem.
wash
Okay, so
solution
I'll
in
just
the
quick
sink.
go through some
Gently
of those
but
things. So first of all, this
firmly
is the
tap the assay
concept of
plate
systems biology. So what
again
is
to
the concept of
remove
systems biology?
residual liquid.
To understand the
Repeat this
total
process
network
for a
interactions
total
that
of
is
4
actually
washes.
happening.
When
So what is
finished,
that?
quickly
Meaning of that?
wipe off any liquid from the bottom outside of the microtiter
So
wells,
any particular
as any
systems and
residue
networks, the
can
metabolic networks, so
interfere
reconstruction of
in
those
the
so that
reading
we can
step.
get and understand our
Make sure
body's
to
mechanistic
not
insights
allow
or any
the
particular disease
wells
or any
to
other condition, we can
dry before
get
the
it. So for
next
that reason, there
step
is
of
use of
adding
technologies like
substrate.
we have like microarrays or those
Immediately after
whatever different
completing the
techniques.
washing
Uh,
process, pipette
uh,
100
and
microliters of
those things,
TMB
whereas
substrate
biology and computation
into
are those
each well.
other things that are involved for the software and to develop new
Then
hypotheses, testing out new hypotheses, testing
incubate
out new
on your
biological questions,
bench
new
at
technologies,
room
new data, new
temperature
insights.
for
So if you
30
see that there are
minutes.
three major domains.
Do not shake
One is
during
biology,
this
biology, then
30-minute
it's computation, the
substrate
technology. So new
incubation
technologies
period,
will lead to new data,
as it may
new
result
understanding will
in
lead to new
higher
biological questions, new insights, and
backgrounds
computation for
and
new hypotheses
less
and those new software.
precision.
So among which maybe we will
Finally,
just briefly
pipette
touch upon other
100
than gel-based and the one-stage
microliters of
techniques
stop
like antibody-based
solution
approaches of which we have seen
into
quite
the
detailed.
wells.
Thanks to them for making this wonderful
With this,
uh,
the
educational videos
microtiter
specifically for
plate is
in
ready for the
terms of matter of our
spectrophotometer.
host cell proteins,
The
of which where
plate
the
should
2D
be
gel and 2D
read
Western
at
blot and new technologies are being developed
450
for
and
the biopharmaceutical
650
industry. So like
nanometers.
ELISA, we have those array membrane. You know that they will have there are three types of
With
ELISA
all
sandwich, indirect
Cygnus
or
products
where you
and
see either
methods,
on
our
the basis of that you are
technical
immobilizing your
support team is available
protein
to
or
assist you
antibody or
in the
antigen.
successful
Like here is an
qualification of
example
our
of antibody
products for
system with
your
ELISA where we have
analytical
antibodies in
needs.
centralized antibody
To
substrate and centralized.
connect with our
Now
experts,
there are
visit
other
us
technologies like the microarrays
online
So for
at
instance, you have the DLMI queries where you can
cygnustechnologies.com.
identify the
Complete
sequence being
our
presented.
technical
Then
support
we have the
form
represent
and will promptly
entire genome of an
respond to
organism, and it
your
helps with
inquiry.
measurement of the gene expression. So the healthy cell, pathological cells, the culture next to the RNA isolation, then gives us the sequencing, and then the hybridization. So what happens is that you have, sorry, so yeah, so you are hybridization between the two strands, you are quantifying it, and then finally capturing high density oligonucleotide arrays,
Hello
uh,
everyone and
multimatrix, and
welcome
this
to today's
cDNA
webinar
uh, microarrays
titled
for measuring the gene
Characterization
expression.
of Residual
Then what where
Impurities
we are
in
moving
Gene Therapy Drug Products,
is that
hosted by Biopharma
protein microarrays.
Webinars
So this
and
protein microarrays,
presented
how
by
this can
Jessica
be used.
Townsend,
So these are
Associate
definitely used for studying
Director
protein
of
antibody interaction, protein
Analytical
protein interaction. So these
CMC
are again
Lead at
immobilization of the captured
Intellia
region, sampling incubation.
Therapeutics.
So these are again
Jessica,
uses
hello
our
there.
fluorescent dyes. So
Welcome.
this
Thank you
also
for
again
joining us
a big
today.
use of our
Hi,
fluorescent
Marcus.
dyes for
Thanks for having me.
having those particular
It's a pleasure.
uh,
And
protein expression levels
following
and to
Jessica, we
identify if
also
there are any
have Eric
specifically
Bishop,
patterns of
Vice
autoantibodies
President
during
of
the cancer detection
Research
or
and
other
Development at
techniques that are
Cygnus
will be useful
Technologies. Eric,
for
hello there,
us.
welcome, and thank you
So
for
now
joining us
moving
as
on,
well.
then we
Thank
have
you,
the
Marcus.
analytical
Thanks for
protein
having me.
microarrays that you can look into it,
It's a
like
pleasure.
functional protein arrays
So I'll be
with
today's
unified proteins with
moderator.
functional
My
domains.
name's
Sorry,
Marcus,
and
and
those are for
before we
studying biochemical
begin, I'd just like
characterizations
to
like PPI,
inform our
protein-gene
viewers
interactions, etc.
that
Then you have
there will
RAPA or
be a
reverse-less protein
live
array, which
Q&A
have different
session
lysate tissue samples
after
which are
both
immobilized and look for
presentations
specific
before
proteins
the
in the sample.
end
So
of the
this is
webinar.
the basic
So
workflow is
audience
given in this
members,
image,
please
which
feel
you can
free
do
to
as well
send in your
as the source,
questions
and those
at any time
particular
via the
publication is
questions
there,
tab
which you can always go
located
back and
directly
refer
below your
to, and you are
webinar
encouraged to do
screen,
so.
and we
So,
will
like
go
these different
through
lysate or
these
tissue samples are immobilized
um,
and look for specific proteins. So, printing samples,
during our
first and secondary
Q&A
antibody
session
incubation,
coming up
light scan,
after
and data
Eric's
analysis.
presentation. And please also
Now,
direct in your
so
questions
this is one of the example of the protein
to
microarrays for
the
autoantibody profiling of glioma
presenter. So if it's a
serum
question
samples to
for
identify
Jessica,
biomarkers
please
using human proteome
mention
arrays.
so
So you
in
can
your
go ahead and
question,
read to
or
understand that
the
how
same
this
for
can
Eric
also
as
help
well.
in diagnosis of
Okay, now please
diseases
allow
like
me to
cancer, specifically which
introduce
kind
our
of brain tumors.
first
So early
presenter.
diagnosis can be a
We have
huge boost
Jessica
to the
Townsend, who
uh,
is
society.
Associate
So these
Director
are some of the
of
technologies apart from the
Analytical
gel-based and mass spec-based
Development
technologies
at
that are also
Intellia
can be used to study and
Therapeutics.
understand the proteome
Ms.
expression
Townsend
uh, at
received
the systems biology
her
level as well.
bachelor's in
So
biology
now if I
from
just
Minnesota
go
State
to some touch
University and
upon
her
to the
master's
concepts
in
of mathematical
molecular
modeling,
and
specifically in terms
cellular
of
pharmacology
understanding when we are
from the
studying the systems biology,
University
that
of
means what kind of
Wisconsin
modeling we need to do
Madison.
for understanding the mechanistic insights, as well as
Following
specifically if you
graduation,
want to
she
design
spent
a
10
particular
years
network,
developing
construct a particular
molecular
network, how to do that.
diagnostics
So
and
first of all, like Boolean
infectious
modeling, which
diseases
uses binary
using
variables, that is 0 or 1, to
PCR
represent the presence or
techniques
absence of biological
and
components.
was
So simplifies
instrumental
these complex
in
systems into discrete states to
several
study
successful
regulatory networks and system
FDA
behaviors. This is for ideal for
F10
qualitative analysis. So this
submissions
I have
prior to
put into it
making the
is meaning of
transition
this
to gene
particular,
therapy.
that which particular
Miss
model will be using
Townsend
for what
joined
particular task, so that it becomes easier
Bluebird
for you
Bio
to go
from
ahead and
2018 to
read about it
2022.
and apply those when you are
Most
doing
notably
or solving any
oversaw the
question for that matter.
residual impurities
Then you have the ordinary
methods
differential equations. So this
utilized in
describes the
testing
continuous change in the system
lentiviral
variables. So these are
vector
the
and
differential
ex
equations like
vivo
enzyme
gene
kinetics, population
therapy
growth.
drug
So all these
products,
systems evolved and how
authorizing
it will be reaching
impurity
those
sections from
equilibrium. Then you have the
3
stochastic modeling when you
BLAs
have
in a
randomness
time
and the probability model system.
flipped.
So
And
this is useful for capturing
recently,
the effects of random
Miss
events
Townsend
on biological
joins
processes like gene
Intellia
expression or molecular interactions.
Therapeutics as
So where
the
deterministic
ex
models may
vivo
fail, that's where we'll
CMC
be using stochastic processes and
analytical lead,
be using stochastic models.
representing
Then is the
analytical
flux balance analysis.
technologies
Specifically, this is
within the
optimization technique
ex vivo
is used
CMC
quite
teams
heavily
across
when I say that it will be
and
for
with
analyzing
that,
metabolic
I'd now
networks,
like to
how much
hand
energy
over to
calculations,
our
how much
first presenter
uh,
today. So,
molecules are
welcome,
required,
Jessica.
and all those kinds of flux balance analysis
Thank you,
to
Marcus.
maximize or minimize an objective function,
Okay,
example growth
so
rate. So how
today
many
I'll be
energy
talking
molecules are
about
required if you want
characterization
to
of impurity
maximize the growth rate,
reduction
provide insights into
in ex
metabolic
vivo
capabilities and
gene
constraints.
therapy drug
Now,
products, as
so I just
Marcus
wanted to give you
stated.
a brief idea about the
My
graphs
name
or the
is
molecular
Jessica
network graphs that you
Townsend,
see,
and I'm
among which
Associate
some of those
Director
meanings
at
of those particular
Intellia.
things like in
And
degree, out
let's get
degree,
this show on the
what is the
road, shall
centrality, so
we?
all those definitions. So among which in degree is something that is the number of
Okay,
incoming edges
so
to
for
a node.
today's
How many
agenda,
nodes are the nodes
we'll start by talking
that
about
are
an
there that regulate
overview
a gene?
of the ex
The
vivo
significance, the
gene
level of
therapy
influence the node gain or
manufacturing
protein
approach.
receives from other nodes. So that particular node
Then
is
we'll
receiving how
move
much influence from
into
other nodes.
what exactly are
High in
residual
suggests that the node is a
impurities
major
and why
target
do
of
they
regulation or
care.
interaction, right?
Following that, we'll talk about CQA assessment and process characterization of
And
impurity
second is the
reduction. We'll
number of
go on
outgoing
to talk.
edges from a node, how many genes are
Actually, the
transcripted and structure
majority of
regulate.
my talk
Significance
will really
reflects
be
the level of
on
influence a
regulatory
node exerts on
guidances
other
on
nodes. So
impurities
high out
and
degrees suggest that the
how
node plays
you
a key role in
use those
regulating or affecting. So you
guidances
can
to
understand that
your
we will be looking at
advantage.
the in degree
And
or
then
out
I'll just
degree.
give a quick
So they
summary
together
of,
provide a activity and
of
roles
my
of
talk.
network nodes, balancing both metrics are required
So
for
let's
the key regulatory
we'll
nodes in biological
start with
networks,
the
and this can
overview
highlight
of
potential
the
regulatory
ex
bottlenecks or
vivo
key integration
gene
points.
therapy
And this is are useful
manufacturing
for understanding network
approach.
dynamics, identifying critical nodes,
So
and
this
studying
is a
functional relationships
schematic
in
of a
biological systems.
very generic ex
So
vivo
this is the
gene
summary. So this
therapy
is to understand
manufacturing approach
variance,
showing a
proteome
CAR T
expression, PPI, PTMs,
example.
etc. All this can be studied
Cells
further
are
in your
removed
systems
from
biology thing.
either the
Yeah.
patient
So
for an
so this is
autologous
all. So
therapy
thank you
or a
all.
donor for an
So if
allogeneic
you have any other
therapy.
question or
They're
otherwise, then
shipped
we
to
can
your manufacturing
end the call.
organization where manufacturing is performed via ex